what is glycolysis? It is there in the name, right? It is glyco plus lysis. So it is a process by which you split a 6-carbon compound called as glucose to form 2 products. And the 2 products that you get differ depending upon whether the process occurs aerobically or the process occurs anaerobically. When the process occurs aerobically, products formed are 2 molecules of pyruvate and 7 molecules of ATP. Let me repeat this. The products of aerobic glycolysis are 2 molecules of pyruvate and 7 molecules of ATP. When the same process happens in the absence of oxygen, products formed are 2 molecules of lactate and 2 molecules of ATP. I am repeating this. When this process happens in the absence of oxygen, products formed are 2 molecules of lactate and 2 molecules of ATP. So if you are asked what is the significance of glycolysis, your answer should be this is the only pathway that can generate ATP even in the absence of oxygen. Because it's a pathway which is not dependent on oxygen, it happens in cytoplasm. Any pathway which is dependent on oxygen should happen in mitochondria. Because glycolysis is not dependent on oxygen, it happens in cytoplasm. So these are the facts that you should know about glycolysis. It happens in cytoplasm. It can generate ATP even in the absence of oxygen. Okay. The next concept that you'll have to know about is pyruvate dehydrogenase complex. What is the function of pyruvate dehydrogenase complex? It is considered as a link between glycolysis and citric acid cycle. Okay, because what did I tell you happens in glycolysis? Just now we learned in glycolysis, from 1 molecule of glucose, we get 2 molecules of pyruvate. What should be the fate of these 2 molecules of pyruvate? They should enter into citric acid cycle. Because what is the function of citric acid cycle? Citric acid cycle is the final oxidative pathway through which every fuel gets oxidized as carbon dioxide. I am repeating this. Citric acid cycle is the final oxidative pathway through which every fuel in your body gets oxidized and is released as carbon dioxide, which can be exhaled out. For example, how is glucose oxidized? Glucose becomes pyruvate. Pyruvate should then enter into citric acid cycle. How does pyruvate enter into citric acid cycle? By becoming acetyl-CoA. So pyruvate enters into citric acid cycle by becoming acetyl-CoA. So which enzyme converts pyruvate into acetyl-CoA? That is pyruvate dehydrogenase complex. So all of you tell me, what is the function of pyruvate dehydrogenase complex? Please repeat it along with me. It converts pyruvate into acetyl-CoA so that it can enter into citric acid cycle. After it enters into citric acid cycle, it comes out as carbon dioxide. That is why I told you PDH complex is a link between glycolysis and citric acid cycle. Okay. Now, where does glycolysis happen? We already discussed glycolysis happens in cytoplasm. Where does citric acid cycle happen? Citric acid cycle happens in mitochondria. When I say citric acid cycle happens in mitochondria, logically, where should pyruvate dehydrogenase complex be present? That should also be present in mitochondria. So, do you understand the compartmentalization? Glycolysis happens in cytoplasm. Pyruvate dehydrogenase complex is present in mitochondria. Citric acid cycle also happens in mitochondria. So that has given you the answer about citric acid cycle. It happens in mitochondria. Otherwise, what else do you know about citric acid cycle? It's a final oxidative pathway through which every fuel comes and gets oxidized, gets released as carbon dioxide, which you can exhale out safely. Okay. Now about glycogen metabolism. The phrase glycogen metabolism includes 2 pathways. One is glycogen synthesis. The other one is glycogenolysis. So what are the two pathways included under this phrase glycogen metabolism, glycogen synthesis and glycogenolysis? Both these pathways happen in cytoplasm. Okay. Then about gluconeogenesis. What is gluconeogenesis? Let me help you understand this in a simple way. Gluconeogenesis is a reversal of glycolysis. I am repeating this. Gluconeogenesis is a reversal of glycolysis. What is glycolysis? We learned this so many times. Glycolysis is a pathway wherein one molecule of glucose is split into two molecules of pyruvate. So what is gluconeogenesis? In gluconeogenesis, from two molecules of pyruvate, we synthesize one molecule of glucose. Right? In gluconeogenesis, from two molecules of pyruvate, we synthesize one molecule of glucose. So what is the first enzyme of gluconeogenesis? The first enzyme of gluconeogenesis is pyruvate carboxylase. This pyruvate carboxylase converts pyruvate into oxaloacetate. Pyruvate carboxylase, like pyruvate dehydrogenase complex, is also present in mitochondria. So I'm repeating this for clarity. Pyruvate dehydrogenase complex converts pyruvate into acetyl-CoA, which is present in mitochondria. Pyruvate carboxylase converts pyruvate into oxaloacetate, that is also present in mitochondria. Okay. Pyruvate dehydrogenase complex links glycolysis with citric acid cycling. Pyruvate carboxylase is the first enzyme of gluconeogenesis. Okay. Then this oxaloacetate comes out of mitochondria, reaches cytoplasm. In cytoplasm, it goes through reversal of glycolysis until it forms glucose 6-phosphate. After glucose 6-phosphate, the enzyme, logical name to the enzyme which can convert glucose 6-phosphate into glucose is glucose 6-phosphatase. So which is the last enzyme of gluconeogenesis? Glucose 6-phosphatase, which converts glucose 6-phosphate into glucose. That glucose 6-phosphatase is present in endoplasmic reticulum. So gluconeogenesis involves multiple suborganelles. The first step, pyruvate carboxylase catalyzed step, happens in mitochondria. The remaining steps happen in cytoplasm. And the last step, which is catalyzed by glucose 6-phosphatase, happens in endoplasmic reticulum. Okay. And please remember, the other name for endoplasmic reticulum is microsome. So if you are asked which is the microsomal enzyme marker, your answer should be glucose 6-phosphatase. Okay, microsomal enzyme marker is glucose 6-phosphatase. Gluconeogenesis involves multiple suborganelles, mitochondria, cytoplasm, and endoplasmic reticulum. That's all about gluconeogenesis. Now about fatty acid synthesis. Actually, before fatty acid synthesis, let's try to learn about HMP shunt. Okay, HMP shunt happens in cytoplasm. But what is HMP shunt? You know, glucose that enters into a cell becomes glucose 6-phosphate. When this glucose 6-phosphate is acted upon by glucose 6-phosphate dehydrogenase, it enters into HMP shunt. For now, just remember this, right? What should you remember? Glucose 6-phosphatase is an enzyme of gluconeogenesis which converts glucose 6-phosphate into glucose. Glucose 6-phosphate dehydrogenase allows glucose 6-phosphate to enter into HMP shunt. And this glucose 6-phosphate dehydrogenase and the entire HMP shunt pathway happen in cytoplasm.